Hi everyone, how's everybody doing? A very good evening. If you've joined, then do drop a hi in the chat box. I would like to meet all of you. So, we're back with the FARRE and this is lecture number 7. If you've joined, do say a hi. All right, so I think we are live. So if you've joined us, drop in a hi and hello, and we are going to start today with the gastrointestinal tract infections. And the thing is, obviously, this is on popular demand. You guys wanted GIT to be covered as well. So in the last year's foray, we have covered the nematodes over here. Okay, so all nematodes, your soil transmitted helminths as well as GI nematodes have been covered in this particular lecture on the Medit channel on YouTube itself. Today, what we're going to cover is all bacterial diarrheas. So, E. coli, Vibrio cholerae, Campylobacter, H. pylori, C. difficile. So, that's 5 bacteria. Then rotavirus, a comparison table of all viral diarrheas with rotavirus, and we are going to talk about the intestinal cestodes, especially the tapeworms, both beef and pork tapeworm, Diphyllobothrium latum and H. nana. Okay, so that is going to cover a major chunk. All that will remain is protozoan parasites and your bacterial food poisoning, which is hardly 3 pages in itself. All right, so this is we are going to cover. It's going to be an intense lecture. We're going to cover major chunk of GIT. After this lecture, I want you to go and I want you to watch this particular array in which I have covered the nematodes. All of the nematodes have been covered over here. All right. Chalo. So if you're ready, if you have joined, drop in a hi and we are going to start very soon. I can see a bunch of you have joined. So let's start talking. Of course, in FARRE, we are going to quickly revise in the format of question answer. You know it by now. So when we are talking about diarrheagenic E. coli, so we specially classify diarrheagenic E. coli separately. Why? Because E. coli can cause absolutely any infection under the sun. So amongst the diarrheagenic E. coli, good evening, we have 6 types of E. coli. Okay, EPEC, ETEC, EIEC, EHEC, EAEC and DAEC. This is how I want you to draw in your answer sheets as well. Okay, you write diarrheagenic E. coli and then you say that enteropathogenic E. coli, enterotoxigenic E. coli, enteroinvasive E. coli, enterohemorrhagic E. coli, enteroaggregative E. coli and diffusely adherent E. coli. These are the different types of E. coli which are present, right? You know that when I say enteropathogenic, so P stands for pediatric. So this is the one which leads to pediatric diarrhea or infantile diarrhea. When I say enterotoxigenic, the T stands for traveler's diarrhea. Okay, so your labile toxin and stable toxin. We are going to talk about it. Then you have the enterohemorrhagic with the EHEC, which is the O157H7, which causes the H for HUS, hemorrhagic uremic syndrome, right? So we are going to talk about all of this. First, you tell a little about the genus. Yes, travelers, correct. First, you tell about the genus, which is it belongs to the Enterobacteriales family. It is a GNB. And of course, because it is a Enterobacteriale, it is going to be gram-negative rod. It is going to be motile. It is going to be catalase positive, oxidase negative, glucose fermenter and nitrate reducer as a rule. All right, it is motile and it has peritrichous flagella. You know all enterobacterials have the peritrichous flagella and the peritrichous flagella is denoted by H antigen. Then important antigens is the O somatic antigen and the H flagellar antigen. And like I had told you, especially the uropathogenic E. coli, the E. coli which causes UTI, in that the fimbrial antigen or the pylori, which are the okay, Talib, share your happy news with us. But let's continue studying up till now at this moment. So, pylori or fimbria are the main adhesion or the main attachment factors of this E. coli, especially the uropathogenic one. Okay, so if you're getting a question on UTI of E. coli from the genitourinary tract, then also you have to write the same things because the organism is the same. And over there, what will you focus on in a UTI question on E. coli? You will focus on pylori or fimbria as being the main antigen, as being the main, attachment antigen. Correct. F1 type. Absolutely correct. Exactly. Right. Then you all remember the MVQ, the plus plus minus minus minus, indole positive, MR positive, VP negative, citrate negative, urease negative. Yes, MVQ plus plus minus minus minus. Okay. Then TSI gives me, it's a lactose fermenter. So all lactose fermenters give me what on TSI? They give me A by A, and E. coli also produces gas. So it is A by A with gas and no H2S. These are the general features of E. coli as a whole, and you have also differentiated. Now you're going to make a nice little table in the answer sheet, and you are also going to write in front of it or draw little sketchy diagrams to show the pathogenesis. ठीक है? तो EPEC, like I said, P for pediatric, it causes the attachment effacement type of lesions, and it is non-toxigenic and non-invasive kind of a disease. It causes P for pediatric or infantile diarrhea. So just simply draw a brush border epithelium, show that the E. coli has attached to it, and it has done what? Attachment effacement. It is non-toxigenic, non-invasive. The bacteria is not releasing any toxin. The toxin then going and attacking the cells, none of that thing is happening. The bacteria is also not entering inside the cell and causing todphod or causing destruction. That is also not happening. Simply, the bacteria is sitting on top of the microvilli, and because of that sitting on top of the microvilli, the brush border is totally displaced. It is effaced. Okay, kharab ho jata hai wo. So that is the only reason. And then of course the absorption is not appropriate from the intestine, therefore it leads to diarrhea. So it's a very simple attachment effacement type of lesion where there are the effaced microvilli because of simply the bacteria sitting on top of the brush border epithelium. Okay, excellent Talib, good to know. Now ETEC, ठीक है, ETEC was T for travelers, T for toxigenic. Okay, so this is going to produce two toxins, LT and ST. LT stands for labile toxin, ST stands for stable toxin. What are they labile and stable against? Heat. Okay, heat. So it is heat stable, heat labile. Labile causes increased CAMP, therefore leading to secretory diarrhea. ST causes increased CGMP, therefore leading to secretory diarrhea. They both cause the traveler's diarrhea. And what is the receptor to which they bind on the enterocytes? They both bind to the GM1 ganglioside receptors on the enterocyte. GM1 ganglioside receptors on the intestine. Who is going to tell me in the chat box which other bacteria binds to the GM1 and leads to increased CAMP? And we are going to read that bacteria today also. We are going to revise it just in the next session. Okay. So which other bacteria has the same mechanism of action? Just that bacteria is much more virulent and the toxin is much more potent. So it leads to extreme profuse watery diarrhea. However, the mechanism of action is same as that of labile toxin. Let me know in the chat box. Right? Yes, correct. Correct, mnemonic. Then we have EIEC. EIEC is invasive E. coli. So over here there is proper invasion. So this was non-toxigenic, non-invasive. This was toxigenic but non-invasive. This is non-toxigenic but invasive. Okay, so it is going to mainly invade through the virulence marker antigen or the VMA, and it causes a Shigella like disease like an invasive dysentery. So this is going to lead to dysentery. Okay, so two strains will produce secretory watery diarrhea, ETEC and the enteroaggregative. Two are going to produce dysentery. This one, the enteroinvasive E. coli, and the enterohemorrhagic, which we are going to read next. Not Salmonella typhi, incorrect. Yes, Vibrio cholerae is the one which has the same mechanism of action as the labile toxin of E. coli. Okay, chalo. Please understand that Salmonella typhi causes typhoid, which is enteric fever, which is a systemic disease. We read Salmonella typhi in the bloodstream infections. The major disease caused by Salmonella typhi is not gastrointestinal disease. Okay, diarrhea का main cause नहीं होता है Salmonella typhi. Salmonella typhi is a main cause of fever. It is not a main cause of diarrhea. Okay, so main cause of diarrhea is Vibrio cholerae. It is E. coli. So these are the main diarrheagenic pathogens. चलो, तो VMA आया, VMA invaded, it broke down the cells, invasion of the epithelium, inflammation, ulceration, and ultimately there is dysentery, which is the most important. ठीक है? Then we have EHEC. What is EHEC? Enterohemorrhagic E. coli. That is O157H7 is the most important strain associated with it, and the infective doses 10 to 100 bacilli only are needed. It has the phage coded Shiga like toxin, Very, very important that it is phage coded or bacteriophage coded. What was our mnemonic to remember the phage coded toxins? A, B, C, D, E. Anthrax, botulinum, cholera, diphtheria and EHEC. These are the ones which lead to, which are phage coded toxins. So EHEC is also phage coded and it is Shiga like toxin. It is also written as virocytotoxin. Virocytotoxin, okay, highly potent. It binds to the GB3 receptors. It inhibits the 60S ribosomal subunit, leading to decreased protein synthesis, therefore cell death. And there is hemorrhagic colitis or HUS, hemorrhagic uremic syndrome, right? So you have a child who had diarrhea. He became fine on antibiotics. After 1 or 2 weeks, he comes back with thrombocytopenia, purpural rash, renal failure, oliguria, nephrotic syndrome kind of picture. What is that? That is hemorrhagic uremic syndrome. ठीक है, चलो जी. AEC is aggregative. This also produces a toxin which is EAST1. So enteroaggregative stable toxin 1. This also functions exactly like the LT of your enterotoxigenic ETEC. ठीक है, so it causes aggregative adherence or the stacked brick pattern of the E. coli on top of the brush border epithelium. So it has stacked itself like a pile of bricks. So very easy diagram to draw. You can easily draw this. Then it leads to persistent diarrhea. However, this can also lead to traveler's diarrhea. So whichever has T in its name will lead to traveler's diarrhea. So it can be the enterotoxigenic E. coli or the enteroaggregative E. coli, right? Then diffusely adherent causes diffuse adherence pattern. Here also there is persistent diarrhea. Here also it is adhering. Here also it is adhering. What is the difference? Over here it's adhering like a stack of bricks and over here it is diffusely adhering to the brush border epithelium. ठीक है? ABCDE is the mnemonic for phage coded toxins. Phage coded toxins, right? So A was anthrax, B was botulinum, C was cholera, D was diphtheria and E was enterohemorrhagic E. coli. These are all the phage coded toxins. That is why with this we have written phage encoded Shiga like toxin, which is the virocytotoxin. Correct hai. C5 deficiency will lead to enhanced effect of all of these infections. Correct. एल्टॉर की बात कहाँ से आ गई बेटा? वो अभी विब्रियो कॉलरा में पढ़ेंगे. होल्ड ऑन, राइट? देन यू विल राइट अबाउट द लैब डायग्नोसिस. लैब डायग्नोसिस में आप स्टूल सैंपल लेंगे. माइक्रोस्कोपी में जीएनबी दिख सकता है, बट दिस इज नॉट अ प्रेफर्ड मेथड. व्हाई हैव आई स्पेसिफिकली रिटन हियर? सो दैट यू डोंट एंड अप राइटिंग gram staining and microscopy as the first point in stool examination. Never write it, okay? For stool examination, we rarely ever do a gram stain because anyways, stool is going to be full of normal flora. So anyways, you will see lots of bacteria. It is very difficult to tell. It is impossible to tell on gram stain whether the E. coli that I am looking at is a normal commensal or a diarrheagenic E. coli. So microscopy is definitely not something we perform right after taking the stool specimen. What do we perform? We perform the culture. So we can do put the culture on McConkey agar, blood agar, and we can also put it on XLD and DCA. But XLD, DCA, however, is majorly preferred for Salmonella Shigella. But usually for stool culture, we put McConkey, XLD, DCA. 3 plates are put. Okay. Then McConkey agar में आपको क्या दिखेगा? You will see lactose fermenting colonies. And for especially for EHEC, we have a selective agar. What is the selective medium? SMAC. SMAC is sorbitol McConkey agar. This is the selective medium for EHEC. And usually when we have selective media, we use Suhas, I am not able to understand your question. Please write the full form of what you're trying to ask and then maybe I can answer you. Right. Like I said, so what usually when we use any selective media, what do we see? We look for fermentation. So it seems like when I'm using sorbitol McConkey agar, then maybe it must be fermenting. But no, it's the opposite case. Your EHEC does not ferment. It is a non-sorbitol fermenter. So it gives me the pale kind of colonies on this agar. So instead of finding the fermenting pink colonies, I find the pale colonies of E. coli. That is something to pay attention to. Okay. So sorbitol McConkey, SMAC is selective media for EHEC. Then biochemicals, you can write the MVQ here and the MVQ is going to be plus plus minus minus minus. And then you can write all the points of Enterobacteriales family also. What are all the points of Enterobacteriales family? Nitrate reduction, yes, it is a nitrate reducer, it is catalase positive, it is oxidase negative, and it is a glucose fermenter. And if it is an enterobacterial OF test, oxidative fermentative test, it is going to show both OF positive, O positive as well as fermentative positive. So Hugh Gleason OF test also. So life becomes very, very easy to remember all the biochemicals of E. coli. No rectification needed. Simply remember MVQ and then simply remember the rule of enterobacterials family. Easily you can write 10 biochemicals for each, for Salmonella, for E. coli, for Klebsiella, for anybody, okay, just by following this particular rule. Need PG, PW method enough, should I? Oh, well, of course, I feel that we are enough. Any app and any resources enough. It depends on you how much you're able to practice. So the resource does not matter. It is your practice at the end of the day which matters. Chalo. Then uske baad, we can also do the ELISA and PCR for the toxin gene detection. Viro cell cytotoxicity can be seen for the virotoxin. See, name is virotoxin. Why is the name virotoxin given? Because it is toxic to the viro cells. So, of course, you can demonstrate that also in the lab. So, lab diagnosis is very, very logical. You just question everything and you get your answers. So, the name of the toxin is virocytotoxin. So, you are demonstrating virocell cytotoxicity. You can do serotyping. Why? Itni der se main bol rahi hu, O157H7. So, how do you find out that it is O157H7? O is the somatic antigen, H is the flagellar antigen. Of course, by doing serotyping only. This is the most important point that you have to write in the lab diagnosis. And AST, which is the automated methods, you can perform Vitek or disk diffusion or MALDI-TOF or anything. Ab dekho, why have I made this flow chart? This flow chart I have made. No. Random guy, I don't know what to tell you for this question. Not that I'm aware of. Okay. So, for lab diagnosis, if you write it in this particular flow, then it is going to be very, very highly scoring. Okay? Write specimen collection, then write culture, then write biochemicals, then write the your molecular methods and antigen antibody detection methods. Then you write your cytotoxicity assays or the cell line cultures. Then you write about serotyping and finally you write about AST. This is the flow in which you should write the biochemical testing. Okay? Then treatment is done how? Oral rehydration. EHEC mein you should avoid antibiotics because there is always an increased HUS risk later on, and or antibiotics only when indicated. Depending upon the AST report you should prescribe. Otherwise, for most toxigenic diarrheas, antibiotics are not recommended. Only oral rehydration is recommended, right? So that finishes the entire diarrheagenic E. coli in the simplest, quickest possible manner. Then we have Vibrio cholerae. Vibrio cholerae kya hai? It is not an enterobacterial. It's a non-lactose fermenter, but it's a sucrose fermenter. This also causes toxigenic diarrhea, ABCD. The C was cholera. This is also a phage-coded toxin. The cholera toxin is a phage-coded toxin. Okay? All of this we have existing knowledge. Now, how to write? Based on the salt tolerance, the Vibrio species are classified into non-halophilic and halophilic. Your Vibrio cholerae is actually a non-halophilic Vibrio because it can only tolerate less than 6% of NaCl. Halophilic can grow at more than 6% NaCl. And how do we remember it? People vacation in Andaman. People vacation in Andaman. Okay. So P, V and A. P is Vibrio parahemolyticus, which tolerates 7% NaCl. Vulnificus tolerates 8%, and finally algineulyticus has maximum salt tolerance or maximum halophilic nature because it is able to tolerate 10% of NaCl. Okay? Yes, it is oxidase positive. Correct. Then we have the Gardner and Venkatraman classification, which is based on the O antigen, and it is divided into O1 or non-O1 groups. Amongst the non-O1 groups, we also have the O139. So it is divided into O1, non-O1 and O139 groups. Amongst the O1, you have two biotypes, the classical biotype and L-Tor biotype. Now, I always tell the students that L-Tor ko you remember like Thor. Yes. So, who was Thor? Thor was a strong guy why? So L-tor is a strong and resistant kind of a biotype. When we will now talk about the differences between classical and L-tor, we will see that L-tor is going to be everything resistant, everything positive, because it is like Thor, okay? And amongst these biotypes, there can further be serotypes. 3 serotypes: Ogawa, Inaba, and Helicobacter. Okay, so if I draw the Gardner and Venkatraman classification and I say that Vibrio cholerae, the Vibrio cholerae further can be O1, O139 and non-O1 slash non-O139. Okay, amongst the O1, you have biotypes. You have biotypes. Two biotypes, classical and L-tor. And both of these classical and L-tor then further can be divided into 3 serotypes. 3 serotypes. So even classical can be Ogawa, Inaba, Helicobacter, and L-tor can also be Ogawa, Inaba, and Helicobacter. Please try to draw it in this type of a flowchart in your answer sheet also. Instead of writing like that, draw it like that. Okay, it will save you time. It will put the point across and examiner will understand everything, that you know that either of them can be any of the particular serotypes. ठीक है? Then general features क्या है Vibrio cholerae के? It is a gram-negative curved or comma-shaped bacilli. It has a single polar flagellum showing the darting or the shooting star motility. What is this called as? The single polar flagellum is called as monotrichous. Monotrichous. Because trichous means hair and mono means single. Okay, so this is the monotrichous flagella. Then biochemicals, like you are saying, oxidase positive, urease negative, sucrose fermenter, indole positive. Okay, so sucrose fermenter and oxidase positivity are the two important things about Vibrio cholerae. Microscopy mein, what does it show me? It shows me the fish in stream appearance. So simply draw these curved bacilli and draw the fish in stream appearance on gram stain, right? Then finally, pathogenesis. Infective dose is quite high. You actually need 10 to power 8 bacilli. It colonizes in the small intestine via the TCP, toxin co-regulated pilus. So everybody is attaching with the help of a pilus. E. coli with the F1 fimbriae and Vibrio cholerae with the TCP. Okay, TCP is the is the gene which codes for this pilus, but the pilus is associated with this, so we are calling it TCP. Cholera toxin is an enterotoxin. This is also a phage coded toxin, and it also has the A and B subunits. A and B subunits, where the A subunit is active subunit and B subunit is the binding subunit. Okay, and the binding subunit binds to the enterocyte, then the active subunit goes inside, and what does it do? It increases the CAMP. Okay, so there is the B subunit binding to the GM1 ganglioside receptors, like I had said. So make the GM1 gangliosides, say that the B subunit binds, A subunit enters inside the cell, and it increases the ADP ribosylation, leads to increased CAMP. There is increased CAMP. Okay, as a result, there is activation of the CFTR chloride channels. Usse kya hoga? There is going to be lots of chloride being secreted out into the lumen, and along with that, the water is also going to come out. And because of this water coming out and inhibition of sodium absorption, there is going to be profuse secretory diarrhea. So ultimately, there is profuse secretory diarrhea. You need not write all of this. You simply draw this kind of a sketchy diagram and flowchart in the answer sheet, writing GM1 ganglioside, B subunit, A subunit, adenyl cyclase, increased CAMP, CFTR, and profuse secretory diarrhea. ठीक है? Okay, yes, it is definitely possible, Naresh. All the best. This lecture is supposed to be for the MBBS second year students. So I will see you in 2 years. All the best. Okay, clinical features क्या है? Rice water stools, severe dehydration, hypovolemic shock. This is the problem with it. Okay, so it is not the diarrhea as such or diarrhea per se which is responsible for high mortality associated with cholera. What is the main reason? It is the severe dehydration which can send the patient into hypovolemic shock. ठीक है? Rice water stools are colorless watery diarrhea with the flakes of mucus, leads to severe dehydration, and hypovolemic shock. Of course, if there is severe dehydration, there is going to be oliguria, muscle cramps, and there is no dysentery and there is no fever associated with this pathogen. Okay. Na to dysentery, na to fever. You're not going to find these two things. So comparison when you do of classical and LTOR, like I had said, remember LTOR as Thor. So everything is going to be positive and resistant except one thing which it is susceptible to. What is it susceptible to? The phage which has its name as LTOR. Okay, it is the LTOR phage. LTOR phage. Phage eats bacteria. The name of the phage is LTOR. So it is going to eat up the LTOR. So the only thing it is susceptible to is the LTOR phage. Rest everything it is positive and resistant. So it is going to resist group 4 phage while this guy is going to succumb. It is going to resist polymyxin B while this guy is going to succumb. It is going to agglutinate the chick erythrocytes. It is going to cause hemolysis of the sheep RBCs and it is going to give the VP test also as positive. Rest everything is going to be negative and sensitive in the classical. Okay. So it is going to cause destruction. So what will it destroy? It will destroy sheep RBCs leading to hemolysis. It will destroy and agglutinate chick erythrocytes. It will destroy the VP test also. So everything it is destroying, resistant and positive. Okay, that is LTOR biotype. Please, please, please draw this differences table to fetch excellent marks in exam. Then lab diagnosis mein you have to collect fresh stool. Why is it important to collect the fresh stool? Because hanging drop is your mainstay of demonstration and diagnosis of your Vibrio cholerae. So to demonstrate darting motility, you have to make hanging drop, okay, from a fresh stool specimen. Transport media is alkaline peptone water and Venkatraman Ramakrishnan medium. Please, please remember this, alkaline peptone water and VR medium. Also what can be used is the autoclaved sea water. Then Monsieur's taurocolate telluride peptone water. Monsieur's taurocolate telluride peptone water, MTT-PW. Even if you don't remember the full name, it is PW, MTT-PW. Chalo. Then selective culture media, TCBS agar, which is thiosulfate citrate bile salt sucrose agar. Okay, please write the full form. And on this, we see the yellow colonies, the sucrose fermenting yellow colonies. Very, very important. So I would suggest that you draw this in your answer sheets, in your exams. Okay, make a green colored agar. Make a green colored agar and then on that you draw the yellow colored colonies. Draw the yellow colored colonies and then write that these are the yellow colonies on the TCBS agar of the Vibrio cholerae. So this is TCBS with the yellow colonies of Vibrio cholerae. Then you draw another agar. Make it a little lightish in color. What is this? This is bile salt agar. Bile salt agar. And write that on the bile salt agar, we see the oil drop colonies. What do you see? We see the oil drop colonies. Better to draw it with a gray. So simply you draw with a pencil. So there you make the oil drop type of colonies on the bile salt agar. So you label it nicely, oil drop on bile salt agar. Okay, draw these two diagrams as well. So you have written the on bile salt agar, you are seeing oil drop colonies, and on McConkey agar, you are seeing the pale NLF colonies. That is also very easy. Pale NLF, so you make an orange colored McConkey or a light pink colored McConkey agar. Actually, this is the better color. This is the McConkey agar. And what are you seeing? You are seeing the yellow, pale yellow colonies on McConkey. What are these? These are non-lactose fermenter colonies on McConkey. So I am making raw, rough diagrams just to give you an idea that you can also make such raw, rough diagrams. It hardly takes 5 seconds in the exam. You should just be aware and confident in order to take up this task in the exam hall. Okay. Next, you have special tests. Which are the two special tests for cholera? One is called as cholera red reaction. Other is called as the string test. Okay. So you can very easily draw the schematic diagram diagrams also of it. What is string test? On a glass slide, you take the bacterial suspension, then you add sodium deoxycholate or bile salts. So in the presence of bile salts, the cell wall is going to break open and the DNA is going to stretch like a string of cheese. So that is called as the string test positivity. What is cholera red reaction? Simply take peptone water, add alpha naphthol, then add KOH, and you see the cherry red color. Even if you don't want to remember alpha naphthol, etc., just simply remember the final red production, which is the cherry red color production. And you can write that in cholera red reaction, we do simultaneous detection of indole and nitrate. Okay, so cholera red reaction is positive and string test is positive. Then you can do dipstick for antigen detection, and AST is not routinely performed, but in certain cases it can be performed as well. That finishes the lab diagnosis. Treatment is of course rehydration, and vaccine can be used in cases of outbreak or endemic areas only. Otherwise there is no antibiotic, no other particular specific treatment for Vibrio cholerae. It is only and only rehydration, oral rehydration therapy. Beta Aishi, you can find the notes on the app in the FARRE segment. All the notes are getting uploaded. Just like you were reading in Nishcha batch, you can read from the FARRE batch on the app. Okay, you will find all the notes over there. What is the pH of alkaline peptone water? pH of alkaline peptone water is more than 8. Okay, more than 7.5 actually. So 7.8 to 8 is what the pH of alkaline peptone water is kept at. Okay, fine. All right. Okay. Cholera gravis, no. Isolation bacteria done. Isolation is what is all the culture that I was talking about. This selective agar on TCBS, bile salt, all of this is isolation. Isolation means culture. Isolation means growing on selective agar. That is what is isolation. Okay. Chalo. Next kya hai? All right. Now we're talking about Campylobacter. Actually, we are talking about the bacter buddies now. Campylobacter, Helicobacter, then we go on to C. difficile, and then we'll be done with the whole bacteria of, your GIT. Chalo, Campylobacter kaun hai? It is a curved gram-negative bacilli which is microaerophilic. The major species are Campylobacter jejuni, Campylobacter coli, and Campylobacter fetus. What are we most importantly concerned with? It is Campylobacter jejuni. Like I said, S-shaped gram-negative bacteria shows the characteristic gull wing appearance on the microscopy. So we will draw it in the lab diagnosis part. It also shows me the darting or the shooting star motility. So always remember two bacteria for shooting star motility. One is Vibrio cholerae and other is Campylobacter. One causes rice watery diarrhea and one causes bloody diarrhea or dysentery. So Campylobacter will cause dysentery. This is also very much oxidase positive and it loves heat. So it grows at 42 degrees Celsius. So we always say Campy loves heat. Campy loves the hot temperatures. So Campylobacter loves the heat. Infective dose is very low. Incubation period is about 5 days and ingestion of unpasteurized milk or contaminated poultry. Okay. So just imagine an S-shaped fancy bacteria which is going on a camping trip and there's lots of little chicken and poultry around and unpasteurized milk. So classically unpasteurized milk and poultry is the answer. What is PBS? I don't understand, Himanshu. I don't understand your question. Chalo, theek hai na? So you will make the gram stain. You will say that it is S-shaped. It also shows me the darting motility. Then what is the pathogenesis? Of course, it is through the contaminated food and milk. Goes to the jejunum because the name itself says Campylobacter jejuni. So jejuni is, it will go to the jejunum. There it is going to cause tissue damage and inflammation, and therefore you're going to see RBCs and WBCs in the stool. It causes inflammatory diarrhea. It can also spread systemically in immunocompromised hosts, and as a complication complication, it can lead to Guillain-Barré syndrome or the GBS. Okay? So intestinally, it will cause inflammatory diarrhea and dysentery, and as an extra-intestinal complication, it is going to cause GBS. It is going to cause Retter's syndrome. What is Retter's syndrome? Can't see, can't pee, can't climb a tree. So there's keratitis, can't see, can't pee, urethritis, can't climb a tree, arthritis. Okay, so reactive arthritis or Retter's syndrome, and disseminated disease which can be seen in immunocompromised people. So please remember, GBS and Retter's syndrome are classically associated as complications of Campylobacteriosis. Okay, Campylobacter jejuni. Then for lab diagnosis, you will do the stool. Take the stool sample, or rectal swab in the Carry-Blair medium can be taken. Why only in Campylobacter we start talking about rectal swabs and we never talk about rectal swabs? Peripheral blood smear आप क्यों करोगे बेटा when we are talking about GI tract bacteria? Just think for a minute yourself. Why will you do a peripheral blood smear? Okay. Correct. Skiros, Campylobacter, Butlers. We're coming on to that. So why are we only taking a rectal swab in case of Campylobacter and not in cases of any other bacteria that we have read? Because this guy is microaerophile. Okay. That is why the rectal swab is a good specimen for this, because you can embed it into this Carry-Blair. You remember Carry-Blair is semi-solid. So you embed the swab inside it so it can get the anaerobic or microaerophilic kind of a temperature or conditions. Okay. Microscopy, you will see the gull wing morphology. You will draw the gull wing very, very easily. You will say that, all right, this is my gram stain background, and over here I can see these gull wing morphology. So just draw like this. Okay, what is this? This is the gull wing morphology on gram stain that we can see. Hanging drop will show me what? It will show me the shooting star. For culture, we need to give it the microaerophilic environment, and for that we are going to use Anoxomat and Macintosh Fields jar. And at 42 degrees Celsius, it is going to give me the colonies. Now, the selective media is Curros, Butlers and Campylobacter. Campylobacter is easy. Campylobacter ka bap or Campylobacter blood agar plate. So it is Campylobacter. Now, it is a disease of the developed world. So the developed world people have Butlers. So you can remember Butler selective media. Now the Skiros is very easy to remember. When you were drawing Campylobacter, you were drawing squiggly lines like this. And when you draw squiggly lines like this, it produces the sound of skr skr. Okay, it says skr skr skr. So when you say skr, you remember Skiros selective medium. So remember the three selective media. Please remember microaerophilic. Please remember 42 degrees Celsius. Please remember dysentery. Please remember poultry and unpasteurized milk, and the three selective media. So it shows me the effusing droplet colonies on the Skiros or the Campylobacter medium, and of course Anoxomat jar is what we are going to use. Treatment is conservative. However, drug of choice is azithromycin if in case it has to be prescribed. Finally, the other bacter, which is Helicobacter, and I always say that Helicobacter sounds like helicopter. So just like helicopter has the wings on its head, the Helicobacter also has tough flagella on its head. So it is Lophotrichus. It is a Lophotrichus bacterium, meaning that it has a tuft of flagella on one single pole. So there is multiple polar flagella. Tuft of flagella on a single pole. So just like Helicobacter, it is helicopter. It is also a curved or S-shaped bacteria, and the mnemonic for oxidase positive is vape bench. The H is H. pylori. So it is oxidase positive. And you know from surgery knowledge that for H. pylori we do the urea breath test. So of course this bacteria is a prototype bacteria of urease positivity. So it is oxidase and urease positive bacterium. Okay. Now, yeah, all of this is not important. This is also a microaerophilic bacterium. So you have the, yes, helicopter, correct. You have the bacter buddies, Campylobacter and Helicobacter. They are both microaerophiles. They need 5 to 10% oxygen to survive. Okay, they are both microaerophilic. So after writing the initial discussion of H. pylori, you know that urease production is the most important pathogenic factor of it. So how does it survive? It's a cancer causing bacterium. It's oncogenic. It leads to gastric carcinomas, but it protects against esophageal carcinoma and Barrett's esophagus, right? Yes, it is hydrochloric acid resistant. That is the major important feature. Yes, K punch is the mnemonic for urease production. Excellent, right? Now, urease production. So it is going to convert the urea into ammonia by secreting the urease enzyme. This ammonia is going to create a cloud of protection around the bacterium. So there is going to be an ammonia cloud. What will this ammonia cloud do? It is going to neutralize the gastric acid present in our stomachs. So the gastric acid has been neutralized. Now the pH has increased. As a result, there is a hormonal shift, right? If it infects the antrum, it is going to destroy the D cells. There will be no somatostatin. The negative feedback loop is lost. Now, the parietal cells are just secreting lots and lots of, increased gastrin, leading to lots of acid secretion, thereby causing a lot of damage and leading to hyperchlorhydria, leading to gastric ulcers. Okay, so there is PUD or peptic ulcer disease or active chronic gastritis which happens in these patients. What are some important other colonizing factors which are associated? Adhesins, oxidase, protease, mucinase, all of these enzymes. And then finally the two major important virulence factors are the VACA and the CAGA. What are VACA, CAGA? These are your oncogenes. They are vacuolating cytotoxin and cytotoxin associated gene A. Basically cytotoxins which will turn the bacterium oncogenic, which is actually responsible for causing the epithelial injury, ulcer production, interleukin release, inflammation, and therefore cancer and metaplasia in that particular cell. ठीक है ना? So this VACA, CAGA are the major genes associated with malignancy, gastric malignancy. ठीक है? So क्या हुआ? Enters the stomach, releases the ammonium cloud. Therefore, it is able to protect itself against acidity. Goes and these VACA, CAGA go and destroy the antral cells. As a result, the negative feedback is lost. Lots and lots of acid production, hyperchlorhydria, leading to peptic ulcer disease and eventually carcinomas. The same thing, if it colonizes the entire stomach, then it also destroys the gastrin producing cells. In that scenario, there is hypochlorhydria. Then it becomes protective for GERD and Barrett's esophagus and protective for esophagus. So remember, H. pylori is problematic for stomach but protective for esophagus. ठीक है? चलो. Now clinical features are gastritis, especially chronic gastritis, the peptic ulcer disease, significant risk of gastric adenocarcinoma. However, it has a protective role against GERD, Barrett's esophagus and esophageal adenocarcinoma. So basically it depends upon which part of the stomach is the bacterium colonizing, that whether it will turn into gastric carcinoma or it will in fact turn protective against esophageal carcinoma. It depends upon which part of the stomach the bacteria is colonizing. If it is only colonizing antrum, it is a problem. If it is colonizing entire gastric mucosa, good news for the esophagus, but highly associated with PUD and chronic gastritis. Chalo ji. Then what is your diagnosis of choice? Here no stool testing is going to help you. So please, for H. pylori, remember to not write the common stool microscopy yada. Please write invasive endoscopy guided biopsies, antral or gastric biopsies, again depending upon where the microbe is attacking, where it is causing the pseudomembranous, okay, no, not pseudomembranous, where it is causing the affecting the gastric mucosa. Then you can do the rapid urease test. Biopsy is placed in the urea containing medium and the pH changes the color. So what is rapid urease test? You're simply taking a piece of the tissue, putting it into urease and it turns pink. So you know that urease or rapid urease test is positive. You can also see the bacteria using the Warthin starry silver stain. It is the most important diagram to draw. Simply draw a yellow background and in that you draw your S-shaped little bacteria like this, because Warthin star is silver. Whenever there is silver means black. So you will see the small curved shaped bacteria against the because of the silver staining, right? Then Urea breath test is the mainstay of diagnosis. It is gold standard because it is non-invasive. Basically, you make the person drink a little bit of labeled carbon wala urea, then he exhales out the carbon dioxide which is labeled. Okay, so you have the urea breath test. Fecal antigen detection can be done and serology for IgG antibodies. So the only role of stool sample in H. pylori is antigen detection. It is not the main specimen you collect for H. pylori diagnosis. Okay, major diagnosis is done clinically, is done endoscopically. Okay, not microbiologically. Then of course selective media is the same as Campylobacter. Same as Campylobacter. So you have Campylobacter or Campylobacter, sclerose and your Butler's medium. Same selective medium is used for this also. And this is also microaerophilic, so it also has to be grown in a Macintosh field jar using an anoxo mat. Treatment में 14 days की treatment होती है, triple therapy or quadruple therapy. Of course, you know it better from pharmacology, where you can give omeprazole, clarithromycin and metro or amoxi. And again, you can give omeprazole, tetra, metro and bismuth sulfide. Okay, this is the quadruple or triple therapy. Now, H. pylori and Campylobacter jejuni, what are the differences? It stays in the stomach, especially in the antrum. This guy stays in the jejunum. It causes peptic ulcer disease. It causes inflammatory diarrhea or dysentery. Here, urease activity is strongly positive, but this guy is urease negative. There is malignancy risk, there is no malignancy risk. Here there is the risk of GBS and Rita's syndrome. It grows as 37, whereas Campylobacter grows at 42 degrees Celsius. This is just to give you a mind map. Okay. Chalo ji. Next kya hai? Next is C. difficile. Clostridioides difficile. Please pay attention. It is not Clostridium anymore. All others are still Clostridium. So it is still Clostridium perfringens. It is still Clostridium botulinum. It is still Clostridium tetani. But the only guy who got a name change is Clostridioides. ये सब याद रहेगा बेटा, just by looking at the video, then trying to actively recall. That is how you can remember it. Okay. There is in MBBS, there are no shortcuts. Okay. But we are trying to put it across in a encapsulated small manner so that you can just revise this itself another time, and therefore you can just reproduce it same way in the exam. Okay, so the only way to remember is watching it again, reading it again. So listen carefully now and then listen one more time and then try to recall in your head. That is how you remember it. Okay, otherwise nobody in this world can remember everything. But then it's MBBS, it's medical science. You got to put in the hard work. All right, chalo. Like I said, Clostridioides. It is a changed name, especially for this guy. Who is this guy? Difficile. Difficile means difficult. He was very difficult to grow. Why was he difficult to grow? Because he's an anaerobe, right? And nobody was paying attention to the fact that it's an obligate anaerobe. Everybody was simply trying to grow it in room air. And it was not growing. So they called him Clostridioides difficile. Okay? It belongs to the genus Clostridium. What is the features of Clostridium? It produces bulging endospores and they are anaerobes, right? Like the tetany has the drumstick and perfringens has the subterminal spores. C. difficile also has the subterminal spores. That is the feature of the Clostridium group. It is significant cause of hospital acquired infection, especially antibiotic associated diarrhea. This is the leading cause of antibiotic associated diarrhea. Spore formation allows persistence in hospital environment. And because of this spore formation, it is also resistant to most disinfectants which are used. So, disinfectants like your hand sanitizer and ethanol, they cannot do anything about him. Okay, if you just sanitize your hands with a hand sanitizer thinking you're protected against C. difficile, I am sorry, you're not. Because of the spore, it is highly resistant. It exists as a non-toxigenic commensal inside our body, right? Everybody tends to keep forgetting it. It is a colonizer. It is a commensal. It does not necessarily always start producing toxins. What happens whenever there is prolonged hospital stay or prolonged antibiotic use? This guy turns toxigenic and starts producing the toxin. As a result, there is antibiotic associated diarrhea or pseudomembranous colitis. Okay, morphology, of course, it's a spore forming bacterium, endospore forming, bulging spore forming with the subterminal spores. How does it act? Basically, there is disruption of the normal flora because of use of broad spectrum antibiotics. That is the whole problem. Because the normal commensal flora keeps the C. difficile low. It tells him to shut up and sit. But now that the normal flora is lost, C. difficile is not able to shut down or sit quietly. It is now going to get excited and cause disruption. Now, which are the antibiotics? CACF, clindamycin, amoxicillin, ceftriaxone and fluoroquinolones. Fluoroquinolones and cephalosporins have very high association with your पेशेंट के सामने कुछ नहीं याद आता है, सब याद आएगा बेटा. You just have to keep revising again and again. Fluoroquinolones and cephalosporins have maximum association with the antibiotic associated diarrhea. Two important toxins are here, toxin A and toxin B. Toxin A is an enterotoxin and toxin B is a cytotoxin. ठीक है, enterotoxin causes fluid accumulation and mucosal damage, and cytotoxin also leads to cellular damage. Basically, both of them are actually going and causing damage to the mucosa, leading to pseudomembranous colitis. Then there are some hypervirulent strains. If you can remember, it's a very high yield thing to remember. If you can't remember, that's okay. The name is ribotype B1NAP1027. If you can remember, it's fine, otherwise it's okay. But just remember that C. difficile is associated with a hypervirulent strain. Yes, metronidazole is given in bacterial infection because metronidazole is the drug of choice against anaerobes. Anaerobic and microaerophilic bacteria पे बहुत अच्छी काम करती है metronidazole. That is why for this guy also you will give metronidazole as one of the treatments of choice. But we are coming on it. Please wait for a little bit. ठीक है? So broad spectrum antibiotic depress normal gut flora, C. difficile colonization, toxin A and toxin B production. This is the most important step of pathogenesis because just this much is not going to amount to anything. Until and unless C. difficile produces these toxins, nothing is going to happen. Okay, so now there will be disease. Clinical features क्या होंगे? Pseudomembranous colitis. There is yellow-whitish plaques of pseudomembrane on the colon. You can draw this simple diagram also. Diarrhea ranges from mild watery diarrhea to severe inflammatory diarrhea. It can amount to any kind of diarrhea across the spectrum. There is toxic megacolon and systemic sepsis in severe cases. Okay. Difference between toxin A and B. Toxin A, like I said, is an enterotoxin. B is a cytotoxin. It increases mucosal permeability and fluid secretion like most enterotoxins. Even E. coli ka toxin was enterotoxin. Even Staph aureus produces enterotoxin. So enterotoxins, they all lead to increased secretions and they all lead to secretory type of diarrheas. But if toxin B starts acting, it starts to kill the cells and leads to cytotoxicity and inflammation. As a result of that, there is cell damage and cell death, which leads to that pseudomembrane or pseudomembranous colitis kind of a feature. ठीक है? This is very, very potent cytotoxin. This is encoded by the TCD-A gene. This is encoded by the TCD-B gene. And this TCD-A and TCD-B is what we are going to detect in the gene expert, which I am going to talk about in just a little bit. You take a fresh stool, you put it in Robertson cooked meat broth. Why? Because it's an anaerobe. All anaerobes, irrespective of the disease, they all go in RCM broth. End of story. The moment I say anaerobe, you should say RCM broth. It is always the right answer. Okay. Selective media, CCFA, cycloserine, cefoxitin, fructose, agar. And what do you see on this? You see the nice, bright, yellow colored colonies on the CCFA agar. Okay, nice bright yellow colored colonies. CCYA also you can remember if you want to. Then you have toxin detection, which is rapid or ELISA. Basically, you're taking the stool sample, you're putting the stool sample in this window. GDH is going to tell you that it is C. difficile, and then A and B lines are going to tell you whether it is toxin A producing or toxin B producing. So you have three important antigens associated with C. difficile. Kya kya? GDH, glutamate dehydrogenase, then toxin A and toxin B. These are the three things you need to detect in order to say with confirmation that this is a toxin producing C. difficile, this patient has antibiotic associated diarrhea. This is the bacterium responsible for forming this pseudomembrane in the colon of the patient. ठीक है, molecular diagnosis में gene expert करेंगे. And what will gene expert do? It will detect the TCDA and TCDB, which are the toxin detection genes. And what is the major advantage of doing gene expert? We have these simple, sasta, easy to do rapid tests. Then why do we want to do gene expert? Because gene expert tells me whether this strain is a hypervirulent strain or not, right? Gene expert helps me detect hypervirulent strains. That is the important thing about gene expert. Three important diseases in which we perform gene expert. One is, of course, tuberculosis, one is C. difficile, and one is COVID-19. These are the three diseases in which we can perform gene expert. So please remember to write gene expert for C. difficile also. So what is C. difficile? What are the important key points? It's an anaerobe. It grows in RCM. It produces toxin A and toxin B. There is a hypervirulent strain, causes antibiotic associated diarrhea. What is the treatment? The treatment is very, very special. Very, very special for this guy. There is conservative treatment and specific antibiotics. Please, oral vancomycin. Vancomycin is not available in oral formulations, but specially for C. difficile, oral formulations have been created. Okay, so oral vancomycin or metronidazole, something which is not available in India but is the only treatment of choice for hypervirulent strains, that is fidaxomicin. It is specifically for the hypervirulent strains. So remember, vanco, metro and fidaxomicin. Okay. Then advanced therapy is FMT, fecal microbiota transplant. You basically take the fecal microbiota from a healthy donor and put it in the form of a capsule and give it to somebody with antibiotic associated diarrhea. So what does that do? That replenishes the gut flora of a person with C. difficile. And where did the problem start in the first place? The first thing to start the whole problem was disruption of the normal flora. So we started the topic from here and we ended the topic there. That disruption was the main problem. That is why we are just replacing and replenishing with good flora. Infection control is the most important thing about C. difficile. There's strict hand washing, like I said, because spores are resistant to ethanol. Spores are resistant to sanitizer. That is why no alcohol based hand rubs work over here. Okay, there is absolutely no alcohol based hand rub policy. Then you should use gloves and disinfectants, and patient isolation is very, very important. Please write the key points in a summary to fetch maximum marks. Spore forming anaerobe, antibiotic associated diarrhea, toxin A, toxin B, pseudomembranous colitis, oral vancomycin, and infection control measures. All right, that finishes bacteria. So many bacteria and so many comments also. Yes, of course, I would suggest that you buy it in second year. Why should you not want to buy it? It is very, very important for concept building as well. Wide range antibiotics. Okay, good job. Can you please tell how? Good marks in practical. Yes. Okay. Questions come in what to. Okay. Right. See, Ashi. So I have in fact made a few videos for surviving the MBBS practicals. So you can watch those videos, and I've been telling always that the first rule to perform well in practicals is to appear confident and to not be haphazard and confused. Examiners don't have the time. They just want to come, they just want to ask you 5 questions and they want to go. They also have a life. So don't try to impress them. Don't try too hard. Don't give yourself that tension of, oh my God, I really have to answer each and every question. Because if you will put that pressure on yourself, you're pushing yourself in a corner, and that is going to adversely affect your performance. Just simply feel that I know what I am doing, I have practiced it, I have read it, I have the confidence, and there is nothing that this examiner can ask me which I don't know. I am telling you, there is nothing that they will ask you which you don't know. If you just sit quietly for a moment, try to reduce your tension, answer will come to you. I am telling you, answer will come to you. It is our anxiety and our performance pressure that is what leads us to do blunders in exam and to not be able to answer the things that we know, right? So you have read everything, you know everything, you have the concept, you have seen the stage, you have seen the demo specimens, you have performed the practicals. There is nothing, even the examiners don't want to ask you hi-fi questions, they want to get it done with. Okay, so just have confidence, be calm and patient. No anxiety is the answer, and not trying to impress is the way to impress. All right, otherwise detailed description, I've done one such video to ace the MBBS vivas. Watch that video. Okay, thank you so much, Sana. Udesh, I could not see your message. I could not see your message, Udesh, so I don't know what you have asked. Shubham. Okay. Acute without an A. Thank you so much. You are acute without an A also. Oh, so everyone's writing that. Okay, thank you. Thank you so much, guys. That's flattering. Now let's go back to studying. Okay. Yes, Ashi, all the best. You will achieve it. Now we come on to the viruses. Okay. What are viruses? The which major important virus is responsible for diarrhea? Rotavirus. And you know, rotavirus is rota hua virus. Okay, it is going to cry all the time. Why is rotavirus rota or crying all the time? Because number one, it got a dhoka. What is the dhoka? Double-stranded RNA. Okay, then why is it crying? Because when you get dhoka, your heart breaks. It has segmented RNA. Then nobody gave him an envelope, so he's just crying in cold. So what is the envelope? What is the blanket? Envelope. So it is non-envelope, double-stranded RNA, segmented RNA genome virus, which is the rotavirus, and it looks like a rotating wheel. That is why it's a rotavirus. And finally, who are always crying other than the virus? The children. So this causes pediatric or infantile diarrhea. So just remember, whenever there's rotavirus, everybody is crying. The patient is crying, the virus is crying, and you are also crying, because this has been asked in the exam. So we cannot help the virus, we cannot help the patient, but we can help you so that you can stop crying now. So how will you stop crying? By reading. So categories are: Reoviridae is the family it belongs to. It is the rotavirus. ठीक है? Non-enveloped with icosahedral symmetry, wheel, rotating wheel-like appearance. So remember, rotating wheel is rotavirus. It has a double-stranded RNA. That is the dhoka it got in life. That is why it was crying. Double-stranded RNA with the 11 segments. Okay, rotavirus has the 11 segments. Then most common cause of severe acute gastroenteritis in infants and children less than 5 years. So they are also crying, virus is also crying. Then pathogenicity, it has many serogroups. A to V serogroups are there based on one particular antigen, which is the VP6 antigen. VP6 is the viral protein, the viral capsid protein. VP6 antigen. And then further the group A is divided. The most important serotype are G and P dependent serotypes. So there is the VP6, which is putting it into A to V groups. Then further group A is divided based on VP7 and VP4. VP7 is giving you the G serotype and VP4 is giving you the P serotype. So this G and P combination is what is going to happen. So the most common worldwide is the G1P8 and the strain used in vaccination is G9P11. ओके, G9P11 का कौन सा स्ट्रेन है? 116E strain, which provides cross protection against G1P8 also. ठीक है? So there is G1P8 and G9P11, two most important strains associated with rotavirus. Who classifies these strains? VP7 and VP4. All right. Then you have a non-structural protein 4, which codes for the enterotoxin, which is produced by the virus. That is going to lead to the watery diarrhea that we are going to see. Right? Of course, it's going to enter inside the cells, affect the cells of the small intestine. Gastric mucosa is never involved. The NSP4, NSP is non-structural protein that acts as a enterotoxin, and as a result there is watery diarrhea associated with the rotavirus. Okay, clinical features: what is the target population? Less than 1 year, abrupt onset of vomiting in profuse watery diarrhea, fever and significant dehydration are the associated symptoms which are seen. Of course, you will collect the I told you, specific receptors of rotavirus are the VPs. It is a non-enveloped virus. There is no envelope. So, there are no spike proteins in rotavirus. So, don't go looking for GP or E or such words. It is a non-enveloped virus. So, a non-enveloped virus will only have the nucleocapsid, and nucleocapsid antigens are called as viral proteins. So, the VP, VP6, VP7, VP4 are the major antigens involved. ठीक है, stool है इसका sample which you will correct. You will do the antigen detection. See, always for viruses, your serology or your antigens or your PCRs are the mainstay of diagnosis, because it is almost impossible to culture or cultivate your viruses. Okay. Then molecular tests are RT-PCR for the RNA detection, and serology is the antibody detection. Treatment is fluid replacement is the mainstay of treatment. Then you have the vaccines. One vaccine which was discontinued due to intussusception is RotaShield. Now what is used is Rotarix and Rotavac. What are they? They are G1P8 or G1P9. This G9P11 116E strain is what is used, and you know that these vaccines are live attenuated vaccines. These are live attenuated oral vaccines given in the form of oral drops. Done and done, right? So there are some other important viral causes of gastroenteritis. You have rotavirus, adenovirus and norovirus. You know, we always say 40-41 is 40-41. Okay, so adenovirus 40-41 leads to diarrheas. Okay, this has a double-stranded DNA like a normal happy or a DNA virus, right? Rota has double-stranded RNA with the 11 segments, causes disease in children, and NSP4 is the enterotoxin which is responsible for causing the disease. Norovirus is also the Norwalk virus. So remember, winter walk or north walk. So this is responsible for most common winter diarrheas in the adults. Okay, these are the important viruses associated with diarrheas. Now we come on to the largest segment of today's lecture, because now we plan to finish 3 important parasites, the entire cestode family. You have the tapeworms. So we are going to talk about the fish tapeworm, Diphyllobothrium latum, the dwarf tapeworm H. nana, pork tapeworm Taenia solium, beef tapeworm Taenia saginata. All of these we are going to talk about right now. So it is going to be a comparison that we are going to read. See, the thing is, you read all 3 in this format, and then whichever one gets asked, you simply write about that one. Okay, so there are some common features about cestodes, there are some common ways in which they present, and that is what we are going to see. Okay, yes, it is possible to revise micro in 2 days. It is not possible to read entire micro in 2 days, right? So please start with FARRE series so that you can form some baseline. Watch the lectures at 2x, try to recall very, very fast, and then try to finish up the syllabus. Okay, chalo. Yeah, so we are going to look at these comparisons. We're also going to look at these very, very high yield DPDX flowcharts. In this, flowcharts itself, we mark and we write everything that we need to remember about the organism. And finally, we are going to have these large tables of comparison, which can really help us visualize and memorize and reproduce in the answer sheet. Okay, na? So that life becomes very, very easy, right? So let's just take in a deep breath. Let's put a smile on our faces, because we are going to finish off cestodes. It is quite large, but we are going to finish it. Chalo guys. So we have pork tapeworm, beef tapeworm, fish tapeworm, dwarf tapeworm. Then some special ones which you don't need to read. The Asian tapeworm, rat tapeworm, and the cat tapeworm. Okay? And you don't need to read them. Simply, you can remember the names. Notes are going to be available on the app. You can access the FARRE batch, which is free. There you can find the notes. You have Taenia solium, which is the pork tapeworm, saginata, beef tapeworm, Diphyllobothrium latum, fish tapeworm, H. nana, Hymenolapis. Please guys, Pay attention to the spellings, because sometimes in answers, like I said, examiners also want to get done with your viva and go. Sometimes they just ask you to write the correct spelling. Please do not write incorrect spellings of organisms. That's a big blunder. That's a sure shot losing out on marks and irritating the examiner. Okay? Please write the correct spelling. So Hymenelepis nana, Taenia asiatica is Asian, Hymenelepis diminuta is rat, and Dipelidum candidum is cat. इनको छोड़ भी सकते हैं. Diphyllobothrium latum is something you don't have to forget. Now, the organism itself is telling you all the features right in its name. Di means two, phyllo means food, bothrium means lips, latum means broad. Okay, so it's a tapeworm which has broad segments. It is a tapeworm with two mouth slits. It is a tapeworm with two intermediate hosts. So Diphyllobothrium latum. Okay, then Hymenelepis nana. Nana is small. Nana says no. So there is no intermediate host. Everything these parasites, they tell in its name only. ठीक है? General features क्या है? Multicellular flat segmented round worms. These are all your tapeworms. Okay, all of them have human as the definitive host, except for whom? Except D. caninum, which anyways you're not going to read. So all of these guys, Taenia, solium, saginata, H. nana and D. latum, in all definitive host is human. No exceptions. ठीक है? Intermediate host usually is one. What are the exceptions to this intermediate host? The one which says nana, no is H. nana. It has no intermediate host. And the one which says di, which is two, Diphyllobothrium has two intermediate host. Rest all, what is remaining? Taenia. Taenia has one intermediate host. Okay, which is either pork or beef, depending upon what tapeworm we are calling it. All right. So, no intermediate host ka exception is no, nana, H. nana. Two intermediate hosts are seen in Diphyllobothrium latum, and Taenia has one intermediate host depending upon the species. In pork tapeworm, intermediate host is pig. In beef tapeworm, intermediate host is cattle, beef, ox or cows. All right. Then egg structure is common feature for all. All cestodes have the hexacanth oncosphere embryo with the 6 hooklets. What is the meaning of this sentence? So all of these tapeworms, they have hooks on their mouth part. So their babies or their larval stages, which are sitting inside the eggs, they are sitting with these hooklets. So just remember, like a baddie larva, baddie baby sitting with 6 knives. That is a hexacanth embryo. H. nana will also have hexacanth embryo in the egg. Taenia will also have hexacanth embryo in the egg. It is only Diphyllobothrium, which I call the Dhakkan of the cestode family. He is a Dhakkan. He wants to be a trematode. So he is going to have an operculated egg. So we will look at it in just a minute. All right. So let's look at pathogenesis and life cycle of all. So I am going to come back to this slide. Before that, let's start looking at these. Okay. So because these are easier to remember, these are easier to understand. So, इससे पहले what was I telling you? What was that game of exceptions that I was telling you? I was saying that all cestodes, meaning all tapeworms, they have one intermediate host, okay, and they have one definitive host. And definitive host is always human or man. End of story. Okay? Amongst the one intermediate host, there are some exceptions. Exceptions. What are the exceptions? The guy which says no or nana, and the guy which says di or Diphyllobothrium latum. What is the difference? The difference is in the intermediate host. Nana has no intermediate host. Di has 2 intermediate hosts. All right. This is the exception that I was telling you. So when I say Taenia, here there is one intermediate host depending upon the species. So pig in pork tapeworm, cow in beef tapeworm. ठीक है, चलो नेक्स्ट हमने बोला लाइफ साइकिल. पहले देखते हैं hymenolithiasis, which is the life cycle of H. nana. H. nana is the most common intestinal tapeworm to infect the humans or the man. Okay, the mode of infection is what? You ingest the embryonated egg. So the mode of infection of all three, in fact, is ingestion of embryonated egg. That egg goes into your intestine, it exists, the adult comes out. This adult is going to sit in your small intestine, lay the eggs, those eggs are going to come out into the feces, you're going to ingest them back. How? Either through environment or because of auto-infection. Auto-infection, okay? So anorexia is the and flatulence is the most important associated features of your H. nana. H. nana, right? So this is a question which examiners like to ask in vivas, because mostly for vivas for all parasites you end up saying that it is diarrhea. Diarrhea is a feature, diarrhea is a feature. But for H. nana, somehow even more important than diarrhea is flatulence and anorexia. So you have to say anorexia as a very important feature of hymenoleupiasis or H. nana. Of course, for the lab diagnosis, what will you do? You will pick up this egg in the stool. So you will do stool microscopy and you're going to see non-bile stained eggs which actually float in saturated salt solution, because two mnemonics, Neha and feather. So Neha, H was H. nana. What is Neha? Non-bile stained eggs. And what was feather? Feather floats. So feather was the mnemonic for all the eggs that float in saturated salt solution. Feather has the letter H. So H. nana also floats in saturated salt solution. Okay. Now let's quickly look at intestinal teniasis before we go back to that comprehensive table. What is happening in intestinal teniasis? You have again ingested what? You have ingested the Cysticercus larva. So for intestinal teniasis, the mode of infection is ingestion of what? Of larva, Cysticercus larva. Why? Because you have eaten undercooked pork and in the muscles always the larva sits. So the special feature of tinea is that the larva sits in the muscles. It can sit in the muscle of human, it can sit in the muscle of pig, it can sit in the muscle of cow. So depending upon what you are eating, you will get the infection. If you are eating pork or beef, you will get the larva from their muscles. When the larva goes inside your body, it is going to convert into an adult. You have ingested the larva in your intestine, the larva is going to become an adult and it is going to stay there for a long time. For 10 to 14 weeks, it is going to start producing the eggs. The segments are going to become gravid and finally these eggs are going to be excreted in the feces. These eggs will be eaten up by the cows and pigs while grazing the ground. Okay, and then you eat them again and the cycle repeats. Now just imagine if instead of this cow, there is a human eating this grass. So of course you will not find humans grazing the grass. But what will you find humans doing? You will find humans eating salads and carrots and potatoes without properly washing. So now human is, if in case human is over here, then human is going to have neurocysticercosis. But when we are talking about intestinal teniasis, in that case, human is here. What is human eating? Pork or beef. And along with that, the Cysticercus larva. So if it is Taenia solium, it is Cysticercus cellulose. If it is Taenia saginata, it is Cysticercus bovis. What are the clinical features? Diarrhea, flatulence, anorexia, anything. Okay. Yeah, the larva crosses. You see, it will go and settle in absolutely any muscle. It can settle in the skeletal muscle, it can settle in the eyes, it can settle in the brain. Right? So if the larval stage, which is very, very small, that can easily, they are microscopic, they can cross, right? Then it can cause neurocysticercosis. Of course, for lab diagnosis, for intestinal teniasis, what will you do? This egg which was coming out in the feces is what you will detect. What will you see on stool mount? You will see bile stained eggs. Bile stained eggs. And actually not just eggs. These small little segments or the proglottids are also shed from the tapeworm. So proglottids and eggs can also be shed in the stool. So you can see proglottids, you can see eggs, and eggs are going to be bile stained. You can detect coproantigen and you can do PCRs. Then let's look at Diphyllobothrium latum. Okay? Diphyllobothriasis. So it is di for dhakkan. It is the dhakkan of the Cestode family. Why? Because he's trying very hard to become a trematode. What is a trematode? There is a general rule for all trematodes. What is that rule? That they all have operculated eggs. They all have two intermediate hosts. Cestodes don't have operculated eggs. They don't have two intermediate hosts. They have one intermediate host. But this Dhakkan right here is a stupid guy. He will have operculated eggs. He will have two intermediate hosts. Anyways, there is di in its name. Okay. So how do you get it? You are here. So what is the mode of infection? You are ingesting with fish. So the mode of infection for H. nana is eating embryonated egg from the environment or auto-infection through your own fingers. The mode of infection for Taeniasis is eating raw beef or pork. So along with that, you're ingesting the larva from the mussels. And the mode of infection for Diphyllobothrium latum is eating the larva because it is present inside the fish. So simply remember the name of the tapeworm. Diphyllobothrium is fish tapeworm. So of course, mode of infection will be fish. Name of Taenia solium is pork tapeworm. So mode of infection is pork. Mode of infection is beef. And H. nana says nana no no. So mode of infection is nobody else but you yourself. Auto infection. Okay. It's very, very simple. Just listen to what the parasite is saying and you will get all the answers. Right. So you have ingested the plerocercoid with the fish. It has entered your intestine. It has become an adult. When it becomes an adult in the intestine, this is the point it will do the clinical features. Now, B12 is something which has to be absorbed from the intestine. This Diphyllobothrium latum. Latum is a very, very big parasite. It is latum. It is broad. It is a huge tapeworm. So it is going to eat up all your nutrition, not leave anything for you. So you will have what? B12 deficiency and megaloblastic anemia. It is synonymous. Even if I wake you up from sleep and I ask you, what does Diphyllobothrium latum cause? You have to utter out B12 deficiency. Even before you say diarrhea, even before you utter any random fact, first thing examiner wants to hear from you is megaloblastic anemia or vitamin B12 deficiency. So Diphyllobothrium is B12 deficiency. D for Diphyllobothrium, D for deficiency. Okay. Now, these adults will lay eggs in your intestine. These eggs will be passed in your stool. You will see these eggs. So, what is the diagnostic stage for all three? Stool. Eggs in stool. What is the infective stage? Larva for 2 and egg for 1, which is H. nana. Okay? Now this is going to come out. Now see, it is behaving just like a trematode. All trematode eggs, they hatch in water and they have many, many larval stages. So it's a Dhakkan. He will also want to behave like a trematode. So it has also released the operculated egg. The operculated egg is embryonating in water. It is becoming Cholerae, procercoid, plerocercoid, and finally ingested by the human. If you can remember Cholerae, procercoid, plerocercoid, good for you. Otherwise, just write first larval stage, second larval stage. Just remember the plerocercoid, which is the infective stage. Okay? In between, it was going through the second intermediate host. So first intermediate host, which is the crustaceans. So first intermediate host for Diphyllobothrium is the crustaceans, the crabs, etc. Then it goes into the fish, and this fish is the second intermediate host, or the paratenic host for this parasite. Okay, stool. Here you will pick up the stool, you will see the operculated eggs or the broad segments which have broken and entered into your feces. ठीक है ना? So this is one go. We have seen the life cycle. In one go, we have seen all the major important points. Now let's compare and contrast. What is the mode of infection for Taeniasis? Ingestion of undercooked pork for Taenia solium, beef for Taenia saginata. Diphyllobothriasis, ingestion of fish containing the plerocercoid larva. Hymenoleupiasis, ingestion of eggs and also auto-infection. What are the infective stages? For Taenia solium, Cysticercus cellulose, which is the larva. For Taenia saginata, Cysticercus bovis, which is the larva. For D. latum, the plerocercoid larva. And for H. nana, the cysticercoid larva, or actually it is the eggs. The eggs. Intermediate host for dilatum, first is crustaceans or cyclops and second is fish, and that's it. GI features diarrhea, bloating, etc. For H. nana anorexia, for dilatum megaloblastic anemia. ठीक है? Taenia solium can cause both intestinal teniasis as well as neurocysticercosis, whereas Taenia saginata only causes intestinal infection. The beef tapeworm can never cause neurocysticercosis, right? Yes, you do, Ashi. Correct. Diphyllobothrium latum leads to B12 deficiency anemia. H. nana leads to anorexia, like I said. Okay, done. This we have seen. Now, okay, sorry, this is a repeat. Right. So here is the comparison of all. Between Taenia solium and saginata, this can be a very important short note in itself. Okay, like a 3 marker. To differentiate between the two. So Taenia solium is the pork tapeworm, saginata is the beef tapeworm. Hooklets are absent in the beef tapeworm. So cows don't have sharp teeth. So similarly, the worm also does not have the hooklets. Rostillum is also not present in Taenia saginata. Suckers are only present in Taenia saginata. And uterine branches are much more in Taenia saginata. How to remember? Remember that cows are like old mothers. है ना? गाय भी हमारी माता है. So mother means has to have lots of proglottids, so many uterus is present, okay, and since cows are humble and sublime, so no hooklets, no rostillum, just simple suckers. Imagine like a old person's mouth, okay, simply suckers are there. That is Taenia saginata. No hooklets, no rostillum, only suckers and more uterine branches in the gravid proglottids. You can also draw very, very simple diagrams. Simply draw this scolex, then draw the neck. Over here, make a rostillum, make the hooklets, make the suckers. This becomes Taenia solium. Over here, draw this guy, draw the neck, simply draw the suckers. This becomes Taenia saginata. Very simple, easy diagrams. Then cestodes have flat segmented body. They are monoecious. They have hexacanth egg. Larval stages are the infective stages and infection is by ingestion. Okay, all of these are the common features. Diagnostic stages: embryonated egg of H. nana and operculated egg of D. latum. Taenia का egg कैसा होता है? Bile stained. It is bile stained with a thick shell and there is the hexacanth embryo inside. H. nana has non-bile stained egg with the polar filament. So we will just draw it. Wait a minute. D. latum has the operculated. So D is the dhakkan, which is the lid of the cestodes, right? Then stool microscopy you're doing concentration techniques and you can do the copro-antigen detection. This is common for lab diagnosis of all cestodes. So no matter which cestode you are asked in exam, write this. Write stool concentration, write egg morphology, write copro-antigen detection. That is why we always read in this form, okay? There is nothing left in this slide. So simply look at this slide and formulate in the form of an answer. Parasitology has to always be read through DPDX charts only. That is the only correct way to read parasitology. ठीक है ना? हो गया. Now let's actually try to draw the eggs. Let's try to look at the eggs. Okay. So this is how you will draw the egg of H. nana. This is how you will draw the egg of Taenia. Now guys, you cannot differentiate between Taenia saginata and solium based on eggs. Okay. Right. And what is this? This is Diphyllobothrium latum, D. latum. So D. latum is clear. It's a large oval operculated egg with the immature embryo inside and a small little posterior knob. So you will draw like an oval egg, you will draw a lid and you will draw a knob and you will make the undifferentiated larva inside. Then you will draw this guy, you will take pencil, you will draw one round circle. What is this? This is the egg shell. Egg shell. Okay. Then what are these? These are the polar filaments. Polar filaments which are coming from the two edges. In the middle, there is this hexacanth embryo. What is hexacanth? With the 6 hooklets. 6 hooklets inside. So you will draw these 6 hooklets like this, 3 pairs. The 6 hooklets. Hexacanth embryo with the 6 hooklets. And what are these? These are the polar thickenings. Polar thickenings. And you will draw it with a simple pencil, gray pencil, because you will write that this guy is non-bile stained. Non-bile stained, right? And how will you draw Taenia? Taenia, in fact, you will color it brown. You will color it brown. It is very much bile stained, right? So this is a bile stained egg. Bile stained egg. This is also a hexacanth embryo. The 1, 2, 3, 4, 5, 6. 6 hooklets inside. Of course, it is a tapeworm. It has to be hexacanth embryo. Then these are the radial striations in the eggshell. And that is the characteristic feature. So you will draw the eggs nicely. You will remember the DPDX chart, and then you will write in a sequence all your answers, right? So we are done. Please read the nematodes from this lecture. Go and watch it right now. All the nematodes have been discussed. Ascaris, strongyloids, hookworm, Trichuris trichuria, and Enterobius vermicularis. All nematodes have been discussed here. Go home and read protozoan parasites. Only 2 important ones you have to read: Entamoeba histolytica and the coccidian parasites. Okay. And finally, bacterial food poisoning, in which you must read about Staph aureus and Bacillus cereus, where Staph aureus is the short incubation period with the super antigen enterotoxin, and Bacillus cereus is the reheated fried rice syndrome. Okay. And then you will be done and done with GIT. Please do not slack off. Please do not leave it for another time. Okay. Don't try to slack off. Please finish it right now. Okay. Albendazole. Right? Done and done. Thank you, guys. So we are done with this session. I think tomorrow we have CNS and then we have miscellaneous. I am not doing genitourinary because genitourinary was covered in all respects in 2025 FARRE. So you can watch that one as well. All right. So thank you so much, guys. Have a very good night. Do finish off the homework right now. Do not slack off, because you will have one very huge unit done and dusted and completed. All right. So thank you so much. Have a good night. I will see you tomorrow. Bye-bye.